Hello, welcome you all to the biology classes. Hope you are all doing well with your classes with all subjects PCMB. In the last class we have discussed about the brief idea about the enzyme or biocatalyst. Okay, so from the last class we came to know that enzyme is a protein. Enzyme is a protein that will enhance the rate of any reaction or that will speed up the rate of reaction. ಅಂದರೆ ಒಂದು ರಿಯಾಕ್ಷನನ್ನು ಸ್ಪೀಡ್ ಜಾಸ್ತಿ ಮಾಡ್ತದೆ ಯಾರು ಎಂಜೈಮ್. ಅದಕ್ಕೇನು ಕರಿತೀರಿ? ಎಂಜೈಮ್ ಇಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ biocatalyst. It means the catalyst, the enzyme which will increase the rate of a biochemical reaction. Increase the rate of a biochemical reaction. Right. So today's class will go deep into the structures of the enzyme, types of the enzyme and the chemical nature of the enzyme in detail. Okay, and in last class we came to know that just like protein, the enzyme also has a primary structure, secondary structure and a tertiary structure. Okay, for example, primary structure, the secondary structure and the tertiary structure. Okay, mainly in the tertiary structures there are some cavities. Or the cavities or the pockets or the crevices. Crevice means in Kannada it is sandhi anta karitiri. Sandhi means the gap between two any things. So these crevices are known as active sites. And in those active sites the substrate will come and bind. In those active sites the substrate will bind. So then only this substrate can form a product. Okay, firstly we should know substrate, nothing but our raw material for any reaction, or it's also called reactant. So for this substrate we need some enzymes. To speed up the reaction that will convert the substrate into a product. And again at the end of the reaction the enzyme remains as it is. Means there is no change in the chemical nature or physical nature of the enzyme. The enzyme remains as it is. Just the function of this enzyme is to convert substrate into product. Right? So the enzyme will just speed up the reaction, will just increase the rate of reaction so that the substrate can be converted into the product. Right? So here for the reaction to occur the substrate has to bind with the enzyme. The question is, where did the substrate bind? Means at what part of the enzyme the substrate is going to bind. Right, we'll just draw a small diagram to understand it better. Imagine it is an enzyme. Okay, so this is the substrate. So now the enzyme, substrate both have to come together, then only the reaction starts. So they have to attach each other. So we'll just write this substrate over the enzyme. Then the reaction starts. So this substrate converts into a product. But the enzyme remains as it is. The enzyme remains as it is, means there is no change in the enzyme at the end of the reaction. This is the substrate. Sorry, this is the product. This is the product. This is the substrate. Okay. So now the question is, what is active site? Question is, what is active site? So this is the place. This is the place. This is the place where the enzyme and substrate bind. Means at this point of the enzyme, substrate will join. So this point is called active site. Okay, mainly this point is called active site. Okay, so one more question barbodu, what is active site? The answer will be any crevices or pockets on the enzyme where the substrate binds is known as active site of an enzyme. Okay, so if any other thing binds at the active site, the substrate cannot bind. We will discuss this at the later classes, even tomorrow classes, right? But for now, just understand active site means any place of the enzyme or crevices or the pockets or cavities on the enzyme where the substrate is going to be bound, right? That's about a active site. So all this process is known as a catalysis. All this process is known as a catalysis. Okay, here the enzyme is called catalyst, biocatalyst, but the process of conversion of a substrate into a product is known as a catalysis. Okay, here we can see two types of catalysis. One catalysis with the help of enzyme, one catalysis with the help of some other chemicals like inorganic minerals, nutrients or any inorganic chemicals. We will now see a brief idea about the inorganic catalysis and enzyme catalysis. ಅಂದರೆ ಒಂದು ರಿಯಾಕ್ಷನನ್ನು ಎಂಜೈಮ್ ಹಾಕಿ ರಿಯಾಕ್ಷನ್ ಮಾಡಿದರೆ ಹೇಗಿರುತ್ತೆ ಮತ್ತು ಒಂದು ಇನಾರ್ಗಾನಿಕ್ ಕೆಮಿಕಲ್ ಹಾಕಿ ರಿಯಾಕ್ಷನ್ ಮಾಡಿದರೆ ಹೇಗಿರುತ್ತೆ ಅದರ ಡಿಫ್ರೆನ್ಸನ್ನು ನೋಡೋಣ. ಓಕೆ, ಫಸ್ಟ್ ಈಸ್ ಎಂಜೈಮ್ catalysis. Enzyme catalysis. Okay, here as we know that enzyme is nothing but a protein. Protein is formed by the formation of a peptide bond by joining of many amino acids along a chain of amino acids is formed. The bond is peptide bond, right? So this bond will be broken down at high temperatures. This bond will be broken down at a high temperature. This process is called as a denaturation. Okay, so enzymes get denatured at high temperature. Enzyme get denatured at high temperature. Denature means denaturation or they get damaged. They get damaged at high temperature. But when we come to inorganic catalysis, it works efficiently even at high temperatures. Inorganic catalysis work at high temperatures. Okay, because there is no biological molecule, there is no biomolecule, just the chemicals are there, so they perform the reactions, they enhance the reactions even at high temperatures. Okay, because of this reason here the enzyme catalysis do not show the thermal stability. Do not show thermal stability. do not show thermal stability, but here they show thermal stability. They show thermal stability, means the reaction is possible, the reaction will be normal at high temperatures. But one provision is there here. If we extract these enzymes, from thermophilic organisms, then the enzymes are active even at high temperatures. Okay, once we extract the enzymes from thermophilic organisms, then the enzymes will be active even at high temperatures. What are these then? Thermophilic means thermo, temperature, philic, tolerative, living, means ಯಾವ ಬ್ಯಾಕ್ಟೀರಿಯಾಗಳು ಆ ಹೈ ಟೆಂಪರೇಚರ್ನಲ್ಲಿ ಇರ್ತವೆ ಅವಕ್ಕೇನು ಕರಿತೀವಿ? thermophilic organism. Maybe bacteria, maybe any other microbe which is living in an area of high temperature. That is called a thermophilic organism. So if we extract the enzymes from these thermophilic organisms, then these enzymes show thermal stability. They will show thermal stability. Okay, so this you should keep in your mind. So just the inorganic catalysis works irrespective of the temperature or the pressure. So it will work efficiently even at a high temperature and sometimes high pressure also. High pressure. But it will not work. The enzyme catalysis will not work because the enzymes, mainly the proteins, will get denatured or damaged. Yen damage agutte? The peptide bond will be broken down due to high temperature normally. But when you extract the enzymes from thermophilic organisms, those enzymes will be thermally stable. They show, the catalysis shows the thermal stability, right? So that's about the catalysis, the enzyme catalysis or the inorganic catalysis. Just pause the video and take your things whatever you want, right? So now we'll move on to the chemical nature of enzymes. Okay, I mean till now we just know that we have learned that enzyme is made of protein. Enzyme is made of protein, but actually not only protein, along with the protein some other chemicals also help to make the enzyme more active. Means enzyme contain protein, but along with protein some other chemicals are there. Okay, those chemicals are known as cofactors. Next concept we are going to deal is chemical nature of enzymes. Chemical nature of enzymes. In our text they have mentioned as cofactors. In our textbook they have mentioned at the end of this chapter as cofactors. But it's better if we discuss at the beginning only. Okay, so chemical nature of enzyme means chemically what actually they are made of. Okay, so here we have two terms, apoenzyme plus cofactor equals to holoenzyme. Okay, so apoenzyme plus cofactor, that will form a holoenzyme. Holoenzyme here nothing but the complete enzyme. It means the enzyme is made of two things. Enzyme is made of two things, means two different types of substances or molecules. What they are? Okay, here apoenzyme is protein part of the enzyme. Protein part of enzyme. Okay, this must be there. ಇದಿರಲೇಬೇಕು. ಓಕೆ, ಒಂದು ಪ್ರೋಟೀನಿಗೆ ಎಂಜೈಮ್ ಅಂತ ಕರಿಬೇಕಾದ್ರೆ, ಆಮೇಲೆ ಎಂಜೈಮ್ ಅಂತ ಕರಿಬೇಕಾದ್ರೆ ದಿಸ್ ಮಸ್ಟ್ ಬಿ ದೇರ್. ಸೊ ಅಪೋಎಂಜೈಮ್ ಇಸ್ ಕಂಪಲ್ಸರಿ. Along with apoenzyme, along with a protein part, there must be some non-protein part. There must be some non-protein part that will make the enzyme more active. That will make the enzyme more active. That part is known as a cofactor. That is non-protein part of the enzyme. Non-protein part of enzyme. Okay, this non-protein part is called a cofactor, right? So this is regarding the cofactors. Okay, this is there anyhow, but we should understand what kind of cofactors will make the enzyme more active. What type of cofactors will make the enzyme more efficient in a catalysis, right? So generally there are three kinds of cofactors. Three kinds of cofactors. First type of cofactor is prosthetic group. Prosthetic group. The second type of cofactor is the coenzyme. Third type of cofactor is a metal ion. Metal ion. Okay, so here don't confuse among the apoenzyme, cofactor, coenzyme, holoenzyme. All are in front of you. Okay, I am telling you, don't confuse among all these. Okay, these may confuse you. Okay, definitely you will get confusion over that. So only I am writing all that in a single. So better look at all these 3, 4. Okay, no problem. You won't confuse here, but they will confuse here. Coenzyme, cofactor, holoenzyme, apoenzyme. Right? Better pause the video. Write this perfectly. Write this perfectly so that you can understand. Apoenzyme, protein part of the enzyme. Cofactor, non-protein part of the enzyme. Complete together will form a holoenzyme. Means the complete enzyme. But in general, usually the cofactors are of 3 types. Means 3 types of chemicals are attached to the protein to make an enzyme. Okay, means these 3 types of cofactors, 3 types of chemicals attached to the protein part to make a complete enzyme, right? So don't confuse among all those 4 items. Okay, so we'll discuss all these 3 in a detail better to understand, right? So protein part, non-protein part, complete enzyme. We'll discuss what is prosthetic group, what is coenzyme, and then what is a metal ion. Okay, so first cofactor is a prosthetic group. A prosthetic group. So prosthetic group is the organic compound which is permanently and tightly attached enzyme. Permanently and tightly attached to the enzyme. For example, if it is an enzyme, if it is an enzyme, this prosthetic group must be permanently attached to the enzyme. Prosthetic group will be permanently attached to the enzyme. So only the enzyme can work properly, right? For example, the enzyme peroxidase. For the enzyme peroxidase or oxidase, they will convert, these enzymes will convert hydrogen peroxide into water plus oxygen. So for these enzymes there is a prosthetic group called heme group. So heme is the prosthetic group for these enzymes which convert hydrogen peroxide into water and oxygen. Okay, so this is the meaning of a prosthetic group. Means any organic group which is permanently attached to the enzyme, which is tightly attached to the enzyme, so that the enzyme will be more active. Means enzyme is catalytically more active. Means the reaction will be more faster. Right? Better pause the video, write the things. Right? Second one, I'll write better its upper words. So for this, heme is the prosthetic group. Right? Second is a coenzyme. Here it's also organic compound, but it is temporarily attached and loosely attached. To the enzyme. Okay, so prosthetic group is permanently attached to the enzyme. It is temporarily attached to the enzyme. Prosthetic group is tightly attached to the enzyme. Coenzyme is loosely attached to the enzyme. Okay, attached to enzyme, that too only during catalysis. Only during the catalysis. Catalysis means enzyme action, meaning the process of reaction, process of enzyme action or enzymatic activity. So this group is temporarily attached, it will go and attach to the enzyme, speed the reaction, again will be removed. Andre enzyme ಗೆ attach ಆಗುತ್ತೆ ಯಾವಾಗ reaction ಆಗಬೇಕಾದ್ರೆ, reaction ಆದಮೇಲೆ separate ಆಗುತ್ತೆ. Alright, so here for example, usually the coenzymes are vitamins. Usually they are vitamins. Means the coenzymes are usually vitamins. We will take some two examples of coenzymes. For example, NAD or NADP, nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide phosphate. Okay, you will study again this in photosynthesis and respiration chapters. They will come in plant physiology. Also, they will come in human physiology, some chapters also. Right. So, for these coenzymes, means for these enzymes, the vitamin B3 is the coenzyme. These are the enzymes. But for these enzymes, there must be a coenzyme. So, that is vitamin B3 is the coenzyme. Nothing but niacin. Vitamin B3 nothing but niacin is the coenzyme for these enzymes. Okay, ಅಂದ್ರೆ ರಿಯಾಕ್ಷನ್ ಫಾಸ್ಟ್ ಆಗಲಿಕ್ಕೆ ಈ ಎಂಜೈಮ್ಗಳಿಗೆ ಒಂದು ಯಾವುದೋ ಕೆಮಿಕಲ್ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ, ಬರುತ್ತೆ, ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ, ರಿಯಾಕ್ಷನ್ ಮುಗಿತಾ ಸಪ್ರೇಟ್ ಆಗುತ್ತೆ, ಅದು ಕೋಎಂಜೈಮ್. ಸೊ ಓನ್ಲಿ ಇಟ್ ಇಸ್ ಟೆಂಪರಲಿ ಅಟ್ಯಾಚ್ಡ್ ಆ್ಯಂಡ್ ಲೂಸ್ಲಿ attached, right? Lastly, the third component of this cofactor is a metal ion. So metal ions like Zn plus, Mg2 plus, Ca2 plus, or Mn2 plus, these metal ions will form coordination bond between substrate and enzyme. For example, this is the enzyme. I have a substrate here. So this is the enzyme, this is a substrate, right? These metal ions will form coordination bonds here. Imagine this is the metal ion. This line is the metal ion. Okay, line is metal ion. So the metal ion form coordination bond with the enzyme and also with the substrate, so that both will attach together properly. Both substrate, enzyme will attach together properly. This is the main aim of that metal ions, means to bind the substrate and the enzyme properly. Okay, so metal ion is lying at the middle, but it will form coordination bond towards substrate, coordination bond towards the enzyme, so that both attach properly, so that the catalysis will be faster. Catalysis will be normal in an active mode, active speed, right? Like this, along with the apoenzyme, protein part, these three non-protein parts also help the enzyme in a proper catalysis. Right? I'm telling you, pause the video, write down the notes. Okay, don't wait for the video, just pause the video and write down the notes. Okay, ಇನ್ನು ಅರ್ಥ ಆಗಿಲ್ಲ ಅಂದರೆ ಇನ್ನೊಂದು ಸಲ ಗ್ರೂಪಲ್ಲಿ ಕೇಳ್ರಿ, ವಿಲ್ ಟ್ರೈ ಟು ಸಾಲ್ವ್ ಯುವರ್ ಡೌಟ್ಸ್ ಅಗೇನ್. ಬಟ್ ಫಾರ್ ನೌ pause the video and write down the notes, just because we have less time, not less time, let us finish our classes so early, so that we can very soon move on to the discussions towards objective classes or NEET classes or CET classes. Okay, maybe by next chapter you will have objective classes. Sir is going to start objective classes also. So better be fast in learning the things, right? ನೀವು ಕಲಿಯುವಾಗ ಸ್ಲೋ ಆದರೆ ಮುಂದೆ ಬರೆಯುವಾಗ ಕೂಡ ಸ್ಲೋ ಆಗಬೇಕಾಗುತ್ತೆ. ಎಕ್ಸಾಮ್ ಬರೆಯುವಾಗ ಬೆಟರ್ ಮೇಕ್ ಯುವರ್ ಸೆಲ್ಫ್ ಫಾಸ್ಟರ್, ಮೇಕ್ ಯುವರ್ ಸೆಲ್ಫ್ ಮೋಟಿವೇಟೆಡ್. ನಿಮಗೆ ನೀವು ಸ್ಫೂರ್ತಿಯಾಗಿ ತಗೊಳ್ರಿ. ಹಾಗಿದ್ಮೇಲೆ ಯಾವಾಗ ಬೇಕಾದ ಎಲ್ಲಿ ಬೇಕಾದರೂ ಹೋಗಬಹುದು, ರೈಟ್? ದಿಸ್ ಈಸ್ ಆಲ್ ಅಬೌಟ್ ದ protein part and non-protein part or cofactors, right? Now we'll discuss about the nomenclature and classification of enzymes. Means how many classes of enzymes are there? Enzyme ಅನ್ನು ನಾವು ಯಾವ ರೀತಿ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತೀವಿ, ಓಕೆ? ಅಂದ್ರೇನು ಅವು ಯಾವ ರೀತಿ ಕೆಲಸ ಮಾಡ್ತವೆ ಅನ್ನೋ ಅದರ ಮೇಲೆ ಅವನ್ನ ಕ್ಲಾಸಿಫೈ ಮಾಡಿದ್ದಾರೆ. ಓಕೆ, ಸೊ ವಿಲ್ ಸಿ nomenclature and classification of enzymes. I'll take a better marker so that I can write better. Nomenclature and classification of nomenclature and classification of enzymes. Okay, so there is we have seen ICBN, right? ICZN, Okay, like that there is also union for biochemistry, International Union for Biochemistry. International Union for Biochemistry. Okay, so according to this committee, based on based on what kind of reaction they are catalyzing, ಅಂದರೆ ಅವು ಯಾವ ರೀತಿ ರಿಯಾಕ್ಷನ್ ಮಾಡ್ತವೆ, ಯಾವ ರೀತಿಯ ರಿಯಾಕ್ಷನ್ ಹೆಲ್ಪ್ ಮಾಡ್ತವೆ ಅನ್ನೋ ಅದರದ ಮೇಲೆ ಅಲ್ಲಿ ದೇ ಹ್ಯಾವ್ ಮೆನ್ಷನ್ಡ್ 6 categories of enzymes. 6 categories of enzymes. Okay, so we'll see those 6 categories. First category is oxidoreductases. Second category is transferases. Third category is hydrolases. Fourth category is lyases. Fifth category is isomerases. Sixth category is ligases. Okay, briefly ಹೇಳ್ಬಿಡ್ತೀನಿ. ಆಕ್ಸಿಡೋರಿಡಕ್ಟೇಸಸ್, ಆಕ್ಸಿಡೇಷನ್ ರಿಡಕ್ಷನ್. ಯಾವ ಎಂಜೈಮ್ಗಳು ಆಕ್ಸಿಡೇಷನ್ ಮತ್ತು ರಿಡಕ್ಷನ್ಗೆ ಹೆಲ್ಪ್ ಮಾಡ್ತವೆ ಅವಕ್ಕೆ ಕರಿತೀರಿ ಆಕ್ಸಿಡೋರಿಡಕ್ಟೇಸಸ್. ಟ್ರಾನ್ಸ್ಫರೇಸಸ್, ಗ್ರೂಪ್ ಟ್ರಾನ್ಸ್ಫರ್ ವಿದಿನ್ ದ 2 ಸಬ್ಸ್ಟ್ರೇಟ್ಸ್. ಅಂದರೆ 2 ರಿಯಾಕ್ಟೆಂಟ್ಸ್ ಕೂಡ ಒಂದು ರಿಯಾಕ್ಷನ್ ಮಾಡ್ತವೆ. ಅಂದರೆ ಈಗ ನನ್ನ ಹತ್ರ 2 ರೂಪಾಯಿ ಇದಾವೆ, ಅವನ ಹತ್ರ 3 ರೂಪಾಯಿ ಇದ್ದಾವೆ, ಏನು ಮಾಡ್ತೀರಿ, ನನ್ನ ಹತ್ರ 1 ರೂಪಾಯಿ ತೊಗೊಂಡು ಅವನಿಗೆ ಕೊಡ್ತೀರಿ. Like that they will just transfer the groups between 2 substrates, group transfer. So transferases. Hydrolysis, addition of water, means breaking the bonds. Lyases, breakdown. They are different, don't compare with this. Okay, both are different, lyases and these are different. Isomerases, we have seen isomers. ಕನ್ನಡದಲ್ಲಿ ದರ್ ಕಾಲ್ಡ್ ಸಮಸ್ಥಾನಿಗಳು ಅಂತ ಹೇಳ್ತೀವಿ. ಸಮಸ್ಥಾನಿಗಳು, ಆಪ್ಟಿಕಲ್ ಐಸೋಮರ್ಸ್, ಪೊಸಿಷನಲ್ ಐಸೋಮರ್ಸ್, ಮೆನಿ ಟೈಪ್ಸ್ ಆರ್ ದೇ, ಜಾಮೆಟ್ರಿಕಲ್ ಐಸೋಮರ್ಸ್. ಸೊ ದೇ ವಿಲ್ ಹೆಲ್ಪ್ ಇನ್ interconversion of the isomers. And lastly, ligases, which will join the 2 different types of compounds or molecules. Okay, we'll discuss this briefly one by one. First category, first type of enzyme, oxidoreductases. Oxidoreductases. Okay, so simply they help in catalyze, means these enzymes catalyze, help in the reaction of oxidation and reduction reactions. Oxidation and reduction reactions. Okay, so we'll see some examples for that. We'll understand better. Not examples, just analogical examples. So we have substrate. Yes, if it is reduced, plus we have another substrate S dash, it is oxidized. So what happens? The result would be S gets oxidized. Lastly, the S dash gets reduced. S dash gets reduced. Okay, so this is how the reaction takes place. For example, there is an enzyme called alcohol dehydrogenase. Example for this category. Okay, this will convert alcohol into aldehyde just by doing oxidation reduction. Okay, so this enzyme will convert alcohol into aldehyde just by the activity of oxidation and reduction. Okay, this is one of the example for that, right? Move on to the next category. Second one, transferases. So simply they catalyze group transfer between two substrates. Group transfer between two substrates. Here artha agodilla. We'll just take some examples. I have one substrate S which is attached with one group G. Okay. Plus we have another substrate S dash. Okay. These enzymes will just transfer this group from S to S dash. ಏನು ಮಾಡ್ತವೆ ಈ ಜಿ ಗ್ರೂಪನ್ನು ಎಸ್ ನಿಂದ ಎಸ್ ಡ್ಯಾಶ್ಗೆ ಕಳಿಸ್ಕೊಡ್ತವೆ. ಸೊ ಲೈಕ್ ದ್ಯಾಟ್ ವಿ ಹ್ಯಾವ್ ಎಸ್ ಸಪರೇಟ್ಲಿ and this G will be transferred to S dash. So S dash G. Okay, just here the group, maybe whatever, any group, any chemical group, organic, inorganic, whatever it is there. So this group will be transferred from S to S dash. Okay, this is how the group transfers will take place between two substrates, substrate 1, substrate 2, S, S dash, whatever you call. Okay, like that this category works, right? Third category, hydrolysis. Here hydrolysis will just try to hydrolyze different kinds of bonds. The hydrolysis will break different kinds of bonds. Okay, they catalyze the hydrolysis of various types of bonds. They catalyze various types of bonds. ಸುಮಾರು ಟೈಪ್ಸ್ ಬಾಂಡ್ಗಳನ್ನು ಬ್ರೇಕ್ ಮಾಡಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತವೆ. ಹೌದಾ? ಯಾಕೆ? Just by adding water molecule. Okay, whenever water molecule is removed, it's called dehydration. Whenever water molecule is added, it's called hydrolysis. Okay, so dehydration ಮಾಡಿದ್ರೆ bond formation ಆಗುತ್ತೆ, hydrolysis ಮಾಡಿದ್ರೆ bond break ಆಗುತ್ತೆ. For example, the bonds like ester bond, like ether, carbon halide, like that many types of bonds will be hydrolyzed with the help of these enzymes, hydrolysis. Okay, we'll see other three categories in detail. Again pause the video, write down the things. Okay, videona pause madri, adana barkodri, right? So the fourth category of the enzyme, after hydrolysis is lyases. The fourth category of enzymes is lyases. Here lyase means breakdown. They will just perform the breaking or transfer of the group, but they will not completely disturb the bond. For example, here, so we have carbon, we have X, we have Y. This is a compound. Okay, so this lyases will not disturb this bond, but just they will keep these two things away. For example, X, Y, and lastly they will keep carbon double bond carbon, double bond, carbon. Just they will take out the things, groups and keep it away. One more thing you remember, firstly they will keep double bonds as it is and they will not undergo hydrolysis. They will not undergo hydrolysis. You should remember. Okay, they do not undergo hydrolysis. Do not undergo hydrolysis. You should remember that. Right? That's about lyases. Lyase means breaking down. Okay. Fifth category is isomerases. Isomerases. You may be knowing about the isomers. The compounds which have same molecular formula but different structure. ಕನ್ನಡದಲ್ಲಿ ಸಮಸ್ಥಾನಿಗಳು ಅಂತ ಕಲ್ತಿದ್ದೀರಿ. Okay. So there are three types of isomers. Many are there. So we will have three: optical isomers, positional isomers, and geometric isomers. Geometric isomers. Okay. So here these enzymes they will help in group transfer between optical, positional, geometric isomers, or they help in interconversion of the different types of isomers. Okay. For example, ಒಂದು ಮನೆ ಅನ್ಕೋರಿ, 3 ಮನೆ ಅದಾವ. ಒಂದು ಮನೆ ಹೆಸರು ಎ, ಎರಡನೇ ಮನೆ ಹೆಸರು ಬಿ, ಮೂರನೇ ಮನೆ ಹೆಸರು ಸಿ. ರೈಟ್. ಸೊ ಮೊದಲನೇ ಮನೆಗೆ ಏನು ಮಾಡಿದ್ದೀರಿ ನೀವು? ರೆಡ್ ಕಲರ್ ಕೊಟ್ಟಿರಿ. ಎರಡನೇ ಮನೆಗೆ ಏನು ಮಾಡ್ತೀರಿ? ಗ್ರೀನ್ ಕಲರ್ ಕೊಡ್ತೀರಿ. ಬಿ. ಮೂರನೇ ಮನೆಗೆ ಏನು ಮಾಡ್ತೀರಿ? ಸಿ ಕಲರ್ ಕೊಡ್ತೀರಿ. ಆಲ್ ಹ್ಯಾವ್ ಸೇಮ್ ಕನ್ಸ್ಟ್ರಕ್ಷನ್, ಜಸ್ಟ್ ಕಲರ್ ಇಸ್ ಚೇಂಜ್. ದೇ ಹ್ಯಾವ್ ಡಿಫ್ರೆಂಟ್ ಕಲರ್ಸ್, ದ್ಯಾಟ್ಸ್ ಇಟ್. ರೈಟ್. ಆಮೇಲೆ ಏನು ಮಾಡ್ತೀರಿ ನೀವು? ಯು ವಿಲ್ ಜಸ್ಟ್ ಚೇಂಜ್ ದ ಕಲರ್ ಆಫ್ ದ ಹೋಮ್ ಎ ಇಂಟು ಬ್ಲ್ಯಾಕ್. Then you will change the color of the home B into red, and then you will change the color of the home C into green. ಆ ಹೋಮ್ ಕಲರ್ ಏನು ಮಾಡ್ತೀರಿ? ಗ್ರೀನ್ ಆಗಿ ಚೇಂಜ್ ಮಾಡ್ತೀರಿ. ಸೊ ಮನಿ ಅದೇ ಉಳಿತಾ, ಏನು ಮಾಡಿದ್ರಿ ನೀವು? ಜಸ್ಟ್ ಆ ಕಲರ್ ಚೇಂಜ್ ಮಾಡಿದ್ರಿ. ಸಿಮಿಲರ್ಲಿ ಹಿಯರ್ ದೇ ವಿಲ್ ಜಸ್ಟ್ ಚೇಂಜ್ ಒನ್ ಆರ್ ದ ಅದರ್ ಫಂಕ್ಷನಲ್ ಗ್ರೂಪ್ ಆರ್ ದ ಸೈಡ್ ಚೈನ್. ಒಂದು ಸೈಡ್ ಚೈನ್ ಚೇಂಜ್ ಮಾಡೋದ್ರಿಂದ ಏನಾಗುತ್ತೆ? ದ ಐಸೋಮರಿಸಮ್ ವಿಲ್ ಬಿ ಚೇಂಜ್ಡ್. ಸೊ ದೇ ವಿಲ್ ಹೆಲ್ಪ್ ಇನ್ ಐಸೋಮರೈಸೇಷನ್ ಆರ್ interconversion between different isomers. You will study more about this in chemistry. Okay, you will be having a chapter called isomerism, isomers. You will discuss there. Okay, last type of enzyme, sixth category, that is ligases. Ligases. Okay, so what are ligases? ಗೊತ್ತಿರಲಿ ನಿಮಗೆ. ligases for cutting, ligases for joining. ಓಕೆ, ಕಟ್ ಮಾಡಲಿಕ್ಕೆ ಇದನ್ನು ಯೂಸ್ ಮಾಡ್ತಾರೆ, ಜಾಯಿನ್ ಮಾಡಲಿಕ್ಕೆ ಇದನ್ನು ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಓಕೆ, ಸೊ ದೇ ವಿಲ್ ಜಸ್ಟ್ catalyze joining or combining of two molecules or compounds. For example, here we have a DNA ligase. We have a DNA ligase. So whenever a piece of DNA is cut, so to join the DNA we will use the DNA ligase. We will study this in second PUC molecular basis of inheritance. Also we will study this in second PUC biotechnology. where we are going to cut the DNA, join the DNA for many processes like genetically modified plants, BT cotton, BT tomato, many things. We will discuss these things in a second PUC in a detail. Just for now, you remember this cutting lysases, joining ligases. Okay. To remember all these six categories, there is a trick called OTHLIL. Okay. So the trick is OTHLIL. Okay. So OTHLIL, just a minute, mic has been under. So remember this word OTHLIL. Okay. OTHLIL here, O for oxidoreductases, T for transferases, H for hydrolysis, L for lysis, I for isomerases, L for ligases. Here we have two L's. Better you remember. First I will cut, later I will join. Andre modulu cut ಮಾಡ್ತೀರಿ ಆಮೇಲೆ join ಮಾಡ್ತೀರಿ, ಅದನ್ನ ನೆನಪಿಟ್ಕೋರಿ. Other things are possible. OTHLIL, you remember the simple sequence of these 6 letters. OTHLIL. So of course only 6 are there: oxidoreductases, transferases, hydrolysis, lysis, isomerases, and ligases. Okay. Remember the term OTHLIL, you can understand, or you can remember the enzymes properly for your examination purposes if the question is come. Okay. So one more thing about this nomenclature is not given that detail in our text. So they have not mentioned the rules of nomenclature. Okay, because there is a perfect rule for that, for the nomenclature, like first digit, some rules are there, second digit, some rules are there according to this committee. So the nomenclature rules are not there in our text. I mean, not needed for our syllabus, PU syllabus. Just they have mentioned clearly about the classification, 6 classes of enzymes, right? So pause the video and take down all the details of the enzymes, right? So this is all about the classification of enzymes. Okay. After this, we will be discussing about the action of the enzyme. I means how the enzyme will act. I mean to convert a substrate into a product. To convert a substrate into a product, what is the procedure of that enzyme? Means what chemical changes are going to be made in the enzyme, in the substrate, or along with both enzyme and substrate to make the process more active. To make the process more active, right? I will just tell you briefly in today's class and I will discuss detail in a next class. Okay. Firstly, enzyme. We have enzyme plus substrate and will form a enzyme substrate complex. Will have a enzyme product complex. Lastly, we have a enzyme and product. Enzyme plus product. So better we'll write in another manner. So enzyme plus substrate will form enzyme substrate complex, that will again form enzyme product complex. Later on we will get the enzyme plus product separately. Okay, so we can enzyme, sorry, it is the substrate, enzyme substrate complex, enzyme product complex. Lastly, we will get enzyme plus product. ಅಂದರೆ ಒಂದು ರಿಯಾಕ್ಷನ್ ಆಗಲಿಕ್ಕೆ ಏನು ಬೇಕು? ಸಬ್ಸ್ಟ್ರೇಟ್. ನಮಗೇನಾಗಬೇಕಾಗಿದೆ? ದ ಸಬ್ಸ್ಟ್ರೇಟ್ ಹ್ಯಾಸ್ ಟು ಬಿಕಮ್ ಅ ಪ್ರಾಡಕ್ಟ್. Substrate has to become a product, right? But for that conversion of substrate into product, we need the help of an enzyme. So firstly enzyme will join with the substrate. Enzyme will join with substrate to form a enzyme substrate complex. Enzyme substrate complex. By many more chemical reactions, chemical changes, the structure of the substrate becomes structure of the product. In this step, the structure of the substrate becomes structure of the product. Then it's called as a enzyme product complex. Lastly, the enzyme will not undergo any chemical changes. Just the enzyme remain as it is, converting the substrate into the product, converting the substrate into the product. This step is very important, where there are many chemical changes taking place in the enzyme substrate complex, where the structure of the substrate will convert into structure of the product. Means the process is going on. Means what? We want a product from substrate. So the main thing will be going on this step between these two complexes. Okay, so we'll discuss all this in detail in the next class, right? Till that, you whatever your doubts you have, just post your doubts in the group. We will try to come back to you with those doubts, right? Maybe tomorrow will be our last class, last video, if it permits. Or if not, we'll try to make one more video for the completing chapter, right? Almost all we are at the end. We'll discuss other things in the next class. Till that, keep learning. Thank you.